especially in the last few decades of last century, by the technological advancements that have been going on in fields like live microscopy, biochemistry, and genetics. But when did it all started? Well, started more than 100 years ago when these two guys, Santiago Ramón y Cajal and Camillo Golgi, two of the fathers of neuroscience, well, they were interested in trying to understand what's the brain made of, what is in that. Because, of course, if you want to understand how it works, you have to understand its structures and the cells that compose the brain, let's say. Camillo Golgi they were both doctors. Camillo Golgi was interested in the anatomy, and he liked chemistry, and he came up with the technique of staining cells in whatsoever tissue in a way that they were becoming very dark, and then if you would cut the tissue and observe under a microscope, you could see these cells in a very, very high detail. This other guy, Santiago Ramón y Cajal, also, as I said, was a doctor, but he was also a good painter, as you could see from his disguisement. And so what he did was that he learned this technique from Camillo Golgi, and then he started to observe the cells, he started to observe the brain, and to make these wonderful tables that came to our days to date. And what they could do basically was to observe the structure of the brain. All they could do was painting, because 100 years ago we had no digital cameras, so we could not take pictures. And they were trying to guess how the brain was working based on its structure. I chose this image, this table, because it's very nice how Santiago Ramón y Cajal drew this region of the brain, which is called the hippocampus. The hippocampus is like a hard drive from our brain, let's say. And if you can see these arrows that he drew here, basically he could guess which was the direction of the information within this concentric circle, let's say, of this part of the brain. And indeed, he was right. This is how the flux of information flows throughout the hippocampus. But how do the information is spread within the brain? So these cells here are neurons. Everyone has heard at least once in his life the word neuron or the word synapses. From Nobel Prize down to my grandmother, everyone knows what a neuron is, more or less. So more specifically, well, this is a very simple schematics. These are two neurons that are somehow communicating each other. Each neuron has basically two main branches that do different stuff. One is called the axon, which is here, which is this one. The axon is an output station for a neuron. And the other branches are called dendrites. In here and here, the dendrites are the input station for a neuron. So when the axon of one neuron connects the dendrite of another neuron, then at that point, there will be a transduction of an electric signal to a chemical signal that will start an electric signal on the other neuron. This thing happens in this particular structure, which is called the synapse. The synapse is very important in the brain, because through synapses, neurons form connections, and synapses are constantly produced and breaking down in the brain. The rate through which synapses are formed basically are important for functions like learning and memory, which makes our brain working, basically. So the terminal part of an axon is called a button, and the input part of a dendrite is called a spine, which looks a little bit like a mushroom. This is a real image of how a synapse looks like under electron microscopy, which is the technique that we use to image the brain. Now, the brain is not simply composed by neurons. Neurons are very important, everyone knows them, and of course, it's the neurons that do the big job, right? But always these two guys, Santiago Ramonica Hall and Camillo Golgi, observing the brain, found out that there were other cells that maybe were as important as neurons, that people don't know that much, these star-shaped cells here. So these cells are called astrocytes. And, well, if you don't know, astrocytes are actually the majority of the cells in the brain. Astrocytes basically take care of making the neurons work properly. So all of the supporting function that the neuron needs to work properly, well, that's the astrocyte that take care of. So I listed a few here, so metabolic support, clearance of neurotransmitter, etc., etc. So, well, this is not very hard to understand. If you think of a brain as a company, let's say, well, what does a company needs to work? It needs managers, first of all. They're not a lot because the work is very specific. They cost a lot of money, but if they do a good job, then the company is profitable and works better. But aside managers, of course, there's a bunch of people that needs the offices to work properly, right? You need people that cleans the offices, you need people that cooks in the canteen, etc., etc. So all of these supporting roles would be the astrocytes. And if you think about that, well, the neurons cannot survive without astrocytes, as a company cannot survive without supporting personnel. So they do a job that is as important as neurons, although they undergo different roles. And in our lab, what we're interested in is mainly the metabolic support, the metabolic and energetic support, which I'm going to talk now. So everyone knows that when you're studying something, let's say when you're thinking a lot, when you're using your brain a lot, then after a while you're going to have a headache, and people tell you that you need sugar, and then you probably want to take a candy so that your headache goes away. Well, this is partially true. Neurons, in order to work, need to intake glucose somehow. Glucose is needed from neurons to be somehow processed and transformed in energy. What's been discovered lately is that neurons does not take up glucose directly, but there is an entire process that goes on through astrocytes that basically are able to take up glucose, to store it into structures that are called glycogen, and then when neurons have to work a lot at the synaptic level, then astrocytes can transform this glycogen into lactate, which is a molecule that you might have heard of. You know, when you work out a lot, then you start to have a lot of lactate acid in your muscles, and then your muscles hurt. So lactate acid is like trash for muscles, but it's very important in the brain, because it can actually be transported to neurons and then transformed in very precious energy in a very efficient way. Now, our interest is to study the ultrastructural localization, of the source of lactate, which is glycogen. Why? Because we can visualize glycogen very well, we can see it very well, and then somehow we can be able to model it in 3D and try to understand where it is localized inside the astrocytes, and this can be important for a number of projects that we are working on in collaboration for modeling brain energetics in particular. So the way we do that, as I said, is that we take electron micrograph using an electron microscope, which is called a 3-view, and let me show you briefly if it works. Yay. So This is a series of electron micrographs. So what this microscope can do is that it can acquire an image of a piece of brain that is embedded in plastic. Then it can automatically cut very thin sections, about 20 nanometers thick, then image the next one, and so on. This way, we end up with a volume of tissue that has been imaged with an image stack. And then what we have to do next is that we have to segment these images, segment the part that we are interested in, and then we can end up with 3D models. This is, guess what, a video that's been done with Blender. So what we're seeing here is a dendrite. This mushroom here is a dendritic spine. The blue guy is an axon, which is an output of a neuron. This red chip is a synapse, which is a part of the which is a part where the receptors are agglomerated. What we are interested in is to see how the glycogen granules are located compared to the spines and the boutons, because these two structures here are the one that consumes more energy during synaptic activation. So, as I said, to do that, we need to do 3D reconstruction of all these structures that we're interested in. This is a very high magnification electron micrograph. This is about 3 microns wide. This very whitish structure that you see in the middle is the astrocyte. And these black dots here that are pointed with the yellow arrow are the famous glycogen granules we are interested in. So we have very high resolution images that can allow us to reconstruct the whole volume that we are imaging. To do that, we use 2 softwares. One is called Trachium2, the other one is called Elastic. These two, in particular, Elastic is a software that allowed us to speed up the procedure. This block of tissue is very complex, has more than 100 structures. So, as you can imagine, it's very time consumptive, but Elastic can speed up a lot, and in about a week, we can reconstruct all of that. So, now the green structure that you see, this scaffold is the astrocyte, and in a while, you will see appearing the glycogen granules, which are these red dots. You can see a cloud of objects that are apparently randomly scattered all around the 3D space, and somehow we want to understand how they are located around spines and boutons. So basically, our task is to find a way to do this job directly in 3D. Of course, you can imagine that you can take each image, you can point at every single object, and then you can just calculate the distance, trying to calculate the distance. But if you want to take into account the Euclidean distance, then you would have to scroll up and down to see what is near each one of these objects. So it wouldn't be an easy task. Plus, these are about, in this stack, 1,000 objects. So it would take a lot of time for the student. So to do that, we came out with a series of plugins for Blender that are really important, because for the first time we are able to use these 3D models in a quantitative way, so we can export meaningful information from these 3D models of a brain. And this is this is really important, because to date 3D modeling of the brain has been used mainly to observe the structure of the brain in a qualitative way, but so far people hasn't been able to do a quantitative analysis out of it, which is a pity, because the quantity of information in the 3D structures of these reconstructions is huge. So somehow we managed to make this process faster, and I will show you a little demo in a while. So this is the add-on that we have designed in our lab. Mainly the work of Dania, which is hiding there. So I'm not a computer scientist. She is, and combining our expertise, we did this really amazing job. We did this amazing publication. So this add-on has a number of features for displaying in a smart way the different objects that we are seeing. We have adapted this for neuroscience, so it can read dendrites, axon, etc., using a very standard notation. But of course, if you're a computer person, then you can modify the way you want. The main task that it performs is, well, I have a sphere, I have a ball, and I want to calculate the distance of this guy and everything that is around it, and the closest one will be the one this sphere is associated with. So not a fancy job, but still it was a very good accomplishment for us. And then once this algorithm runs for all the objects, you can export the data into a spreadsheet that, for further analysis. Now I want to show you something else. Again, as I said, these models are really complex, so they're not easy to observe. And well, most of you are familiar with 3D modeling, so you know that it's very nice to have a very big screen, right? Because you have a very wide view of what you are observing, and this is the case for us as well. But we were thinking, with the resources that are available in our university, that we might be able to do a better job. So let me show you a little bit. This is a brain of a rat. This bluish thing is the hippocampus, and then we can dive in the hippocampus and start to reach. Well, this is the model that I was showing you before. So you see that no matter how small it is, the quantity of objects that it includes is really huge. Again, as I said, it's more than 100, and it's still not even a very big piece. So how can we use this model to do a more meaningful quantitative observation? Because we had the impression that there was some kind of patterning in the glycogen distribution. So in KAUST, we have this amazing virtual reality installation, the cave, and we have the capability of projecting the Blender viewport into the cave. So this was me 7 kilos ago, and it was one of the first time we were using this installation, and it was very nice. It was really amazing because it's really like stepping inside the brain. This object is about 500 nanometers, and all of a sudden is just this big in front of you. So this thing is not only cool, but the very good thing was that by observing this cloud of objects that were in front of us, we managed to see that there was some kind of pattern in front of our eyes. We could see that they were clustered somehow. So we had the idea of modifying our add-on for clustering. So what we can do is that we can read the position in the 3D space of each granule, and then we can use a clustering algorithm, dbScan, that can take up the position of each one of these objects, and then it can create clusters. So this comes handy also for this task. Then, well, we have added some nice features. So each cluster have its individual color. It's wrapped around an ellipsoid, and then, of course, we can calculate the distance between each cluster and its closest boutons or spine. And the nice thing was that the distance between these clusters matched nicely with the distribution that we have calculated with the single granules. So if I have some time, I don't know, maybe I'm running late. I don't know. I can? Okay, well, I just wanted to show you briefly the add-ons in Blender. If it works. So we have to start Blender through this little script that we did, because some of the functionalities that we use requires Python 3.3. So we had to install Python 3.3 and then readdress the libraries there. So this made our life easier, because I mean some of the functions can be just called through Python, which was really handy. One thing is that this add-on is very strictly linked. Does this work? Yes. Yeah. So this add-on is very strictly linked to Neuromorph, which is the previous one that we have designed, that you can see from here. So I'm going to show you some Neuromorph before. And this because basically what our add-on glycogen analysis does is that it needs to read the position of spines and buttons that has been pre-segmented using Neuromorph before, and this is of course very important. So once that's done, then glycogen analysis can work. So let me show you briefly Neuromorph. Okay, now here I can just activate the visualization. This is glycogen analysis, so we can show just one class of objects if we wish. For instance, here the epithelial cells. There are a number of handy functions that we have implemented with Neuromorph, because one of the driving force of our work was that we were doing a lot of segmentation before, and we needed some tool to proofread the segmentation. So one nice thing that Neuromorph can do is that it can superimpose nicely the original image stack to the 3D segmentation. Then we can do things like scrolling the image up and down. We can also, while thinking that everything has been segmented, show you an object which is in a particular position. So let's say I am interested, so there's a synapse here, as you can see. Let's say I'm interested in this particular axon here. I can click on a point, I can click on display object at selected vertex, and if, oh, of course it's not working. That's demo effect. It was working this morning, so it should show. No, it's not. I'll try one more time. If it doesn't, I'll give up. Okay, it doesn't. Well, it should work. It used to work, but I have modified this file for a video, so I must have changed the relative location of the object. So, well, never mind. Once we use Neuromorph to do the analysis, so Neuromorph basically produces three kind of measurements: lengths, surface areas, and volumes. Yeah, I will cut it. Then I can just show you briefly the glycogen cloud that is here. So these are all the glycogen points. I can just click on a button, calculate the nearest neighbors for each one of these objects. This take no more than 30 seconds, I promise. And I will end up with the whole measurements. I will end up with a little window that can list all the objects that has been accounted, and how many glycogen granules per object are associated. And then the cool thing is that I can just export everything for analysis. Again, this is really a powerful thing, because there's not such an analysis tool in literature, and also Blender has such a good capability of being customized that it's been ideal for our work. So you can see here that we have a list of objects, and for each object I can click on it, I can see how many granules are associated for each one, and etc., etc. So this is really good. Well, I guess I will just finish here, leave you with a little thought. This is an image I like a lot. This is staining of brain cells, and these are galaxies. So two completely different scales. Still, you can kind of recognize that there are some common patterns. So maybe by studying and trying to decorticate the secrets of our brain, we would be able one day to understand better the universe or vice versa. Thank you very much.